Okay, hello everyone again, and welcome back. So we're just moving into the last part of our afternoon session today. So we have a final talk, another discussion, and then a final set of breakout sessions. But it is my real pleasure to introduce our next speaker, who is Professor John Rogers from Northwestern University, who's a physical chemist by training. He's received many best paper awards, career awards, and we're really honored to have him join us today. And Professor Rogers' group seeks to explore and exploit interesting characteristics of soft materials. So I think it's really nice that it gives us a contrast to all the previous plenary talks today. So John, thank you so much for joining us today. Really appreciate it. Apologies for the mix up on the time, and I'll hand over to you.
Yeah, hey, thanks for that introduction. It's a real pleasure to be here. I'm going to go ahead and share my screen and get started. So I was able to jump into part of the discussion that was happening just prior to the break and heard some fascinating thoughts were shared around these xenobot systems. And I'm aware of the research within that community, not directly involved in it, but maybe working on technologies that are complementary or maybe synergistic with that kind of research, specifically, efforts to develop technologies that can coexist with living biological systems on a chronic basis to allow for two-way information exchange and control. And so I'll spend the next 40 minutes or so talking about our work in that broader space, focusing on 3D electronic microsystems and mesosystems and their ability to form interfaces to neural tissues and as the basis for creating biohybrids that might represent an interesting additional capability that could be brought to bear to this notion of xenobots. And so I'll start with a background, a little bit of motivation, give you some sense of how we create these 3D mesosystems and how we can integrate state-of-the-art electronic functionality, microfluidic functionality, optoelectronic components directly into those frameworks, and then leverage them to establish two-way interfaces to living neural tissues and organoids, and then conclude with some initial work that we've been pursuing on biohybrid robotic systems and bio-inspired devices of various sorts. And so, as you might guess, my expertise is in hardware. And we work closely with biological collaborators to understand how to blend advanced hardware with living tissues in novel ways to open up new opportunities that range all the way from systems that could advance the way we think about human healthcare, all the way into new robotic systems. And so my background is in physical chemistry and material science. That's my home department, but I have affiliate appointments in most engineering departments here at Northwestern, but also with the medical school. So I've paid appointment neurological surgery and dermatology also. So we're very closely coupled in the medical community, thinking about opportunities at the interface between engineering science and medical science. And so the overarching goal that we've been thinking about and working toward over the last 10, 15 years is this notion of biocompatible electronics, so electronics for the human body. So thinking about the human brain in particular as biology's most sophisticated form of electronics, and trying to develop classes of man-made electronic systems that offer capabilities comparable to those in state-of-the-art silicon CMOS platforms, but engineered to allow for their intimate integration with the brain for chronic periods of time, either on the surface or down into point contact location at the depths of the brain, or distributed across volumetric spaces, as I'll describe in the presentation today. And if you could do that, you might be able to develop tools for understanding basic processes by which the brain operates, and maybe deliver engineering forms of therapy to address mental and brain disorders of various sorts. And so that's kind of the starting point, but you could imagine that if you could develop a technology that could blend and integrate with the brain, you might want to leverage that same sort of technology as interfaces to other vital organ systems of the human body to extend lifespan and enhance health outcomes. And so So we worked on similar types of devices integrated with the cardiac system. You can imagine many opportunities there. The heart is electromechanical system, being able to map electrophysiology, maybe deliver spatiotemporal patterns of stimulation to the surface and the depths of the brain might be useful in extending the health of the cardiopulmonary system, but not just the heart, but many other points of interface as well have drawn our interest: spinal cord, peripheral nerves, liver, kidney, lungs, diaphragm, many places. And the skin is a nice starting point because it offers a relatively non-invasive point of integration and an ability to deploy on human subjects on a near-term sort of time frame. And so the challenge in realizing that goal is fairly obvious, and it's most clearly illustrated by just thinking about the engineering principles that serve as the foundations for all man-made microsystems. They're constructed out of largely rigid classes of materials: silicon, silicon dioxide, gallium nitride, these kinds of materials, spectacular performance, and an incredible depth of scientific understanding around the properties of those materials. But that rigid mechanics and the planar designs in these devices make it very difficult to integrate with soft, time dynamic living tissues. So whether it's integrated circuits or microfluidic lab on a chip platforms, microelectromechanical systems, optoelectronics, planar photonics, they're all in these rigid, static, multi-layered, but largely two-dimensional types of configurations. And if you look at the biological world, the engineering principles are much different at a very qualitative level. So you think about neural circuits that establish functional capabilities in the brain, or you think about microfluidics in the biological world as embodied by vascular networks. They're intrinsically curved, they're non-planar, they're mechanically compliant, they're time dynamic, and they're intrinsically three-dimensional in these open network type of architectures. And so the problem statement really is, how do you go from a technology like this, the silicon CMOS integrated circuit, and reformulate it to allow for a deployment in, on, or around soft living tissues with chronic stability and no adverse impact on the biology, to establish, again, this kind of two-way communication channel and sensing and therapeutic stimulation, for example. And so we've been working that space for probably 15 years now. There are kind of three streams of effort that we have in that overall direction, as illustrated with the three images that you're seeing on this slide. Starting from the left, we've developed stretchable, skin-like forms of electronics with all the kinds of sophistication and function that you would see in microprocessors used in consumer electronics, gadgetry, but designed with skin-like physical properties and built with biocompatible materials to allow minimally invasive, sort of imperceptible levels of integration on the outside surfaces of the body, the skin, but also surfaces of internal organs. So that's one direction where we have good momentum. The second is illustrated in this middle frame, which is the notion of physically transient forms of electronics, bioresorbable electronic circuits. So you can think of them almost like a resorbable suture, but now with function that expands beyond just simple mechanical fixation to full digital electronic circuitry and radio frequency communication and digital sensing capabilities. And so we've established a portfolio of materials that allow for that unusual class of electronics technology, uniquely defined by its ability to dissolve in a controlled way when exposed to biofluids to address temporary needs in biomedical implants. And then the third is illustrated on the right, and that's what I'm going to focus on today, is really kind of an exploratory effort to think about integration with biology beyond surfaces, which are afforded opportunities associated with the technologies in the left and the center frames, of now thinking about how to expand that mode of interface beyond 2D surfaces into 3D volumetric spaces to create these kind of open network architectures that resemble the neural circuitry and the vascular networks that I mentioned previously. They're ubiquitous in biology, but doing that with the most sophisticated forms of optoelectronic materials to allow for that kind of integration and two-way communication. So I'm just going to mention a couple of quick ideas as background and then launch into the basic principles by which we create those 3D frameworks and how we can use them to integrate with biology. So if you think about silicon, most people think about that material, at least in the context of electronics, in the form of a wafer, which maybe has a millimeter or so in thickness. And at that thickness, with the intrinsic mechanical properties of silicon, you end up with a piece of material that's rigid and brittle, much like a plate of glass. So if you drop it on the ground, it'll shatter into a million pieces. It's processed in a planar geometry to align with the kind of photolithographic techniques that are used for manufacturing in the semiconductor industry. So all sorts of physical properties that are profoundly mismatched from those of soft biological tissues. But you can overcome those limitations with a couple of very simple ideas in mechanical engineering and material science. The first one of which is kind of illustrated on the left here, which is that if you make any material thin enough, regardless of its intrinsic mechanical properties, it will become very flexible and bendable, just because the flexural rigidity scales down with the thickness of the material. So we figured out how to take a silicon wafer and shave off of its near surface region ultra thin sheets, ribbons, wires of silicon, device grade silicon, thick enough to support the kind of electronic functionality you might ultimately want to achieve, but in geometries that are thin enough that allow for this mechanical flexibility. So with that kind of idea, you can integrate silicon with a sheet of plastic and build a very high performance flexible piece of electronics, which is one step in the right direction, but biological tissues not only flex, but they stretch. And they can encompass non-Gaussian surfaces. So really demanding something beyond just mechanical flexibility. And the way you do that is sort of illustrated on the right. You take these very thin strips of silicon, you integrate them with an underlying low modulus elastomeric polymer, but you integrate them in a wavy geometry. And by doing that, you essentially create an effective accordion bellows out of the silicon. So this hard, soft materials composite, the silicon and the silicone, in this particular example, affords an end-to-end effective stretchability with a modulus that can be precisely matched to the properties of a tissue of interest for integration. So really simple ideas. You can extend them, add all sorts of engineering elaborations on top of them. You can exploit that kind of out-of-plane, wavy type of shape, as well as in-plane sort of filamentary serpentine type designs. You can exploit those ideas not only in sort of one-dimensional strips, but two-dimensional mesh constructs. And you can embed high modulus materials and functional components within the framework of that kind of mesh, such that when it's integrated with a low modulus elastomer, in this case, a silicone polymer with a modulus about 50 kilopascals, you can create a deterministic hard soft composite material system designed with insights developed by 3D finite element modeling and the mechanics to create an artificial system that has stress strain properties precisely matched to those of a tissue at a point of interface of interest. And so in this particular case, we developed a skin-like platform modulus comparable to the skin, even though the system includes silicon, which has a modulus of 150 gigapascals, so a million times stiffer than skin, but you can create an effective silicon system by using these geometrical ideas and concepts in hard soft composite engineering. So you can do that. You can create these platforms. They also have the ability to conform to complex topographies presented by biological systems. And this is a scanning electron micrograph colorized to sort of illustrate the ability of these kind of filamentary mesh structures to conform to the surfaces of skin. In this particular case, is a polymer replica of skin, to just give you a sense of how that works. So you can put it all together. It all works. We thought we had a pretty good handle on it in 2011, got a little bit better in 2014. And then starting in 2015, 2016, really geared our efforts around how you can exploit this novel skin-like form of electronics to address unmet needs in clinical care of various patient populations, where our focus is primarily on vulnerable populations, maternal, fetal, neonatal, pediatric health. And so I won't say anything about it, but we've advanced that technology to the point of FDA approvals deployed in 20 countries, and forms the basis of a startup company with partnerships with Draeger, GE, Space Labs, and others who supply neonatal intensive care units with traditional hardware. So adding these kinds of devices eliminates a lot of the burden on the patients, the premature babies in those cases. So that skin, you can take the same ideas as I mentioned in the outset, and you can exploit them for implantable electronics with advanced functionality far beyond anything that's possible with implants as they exist today. And so on the left frame, you see a piece of electronics with the kinds of designs that I described previously resting on the surface of the brain. This is a feline model of epilepsy, so we're monitoring electrical activity associated with seizures that occur in those animal models, just as an example. And then on the right, it's sort of a cardiac interface. Here we have essentially like an instrumented pericardium. So it's a very thin elastic membrane that wraps the entire epicardial surface of the heart and delivers to the surface of the heart various sensors and actuators. So we're monitoring electrophysiology, perfusion, and then we have cellular scale LEDs that can exploit techniques in optogenetics for optical-based pacing of the cardiac rhythm. So those are the ideas. Now, if you think about, as I mentioned before, sitting on the surface of an organ is of interest. And if you can do that in a way that's completely non-invasive and stable over chronic time periods, you open up a lot of interesting applications in as tools for basic biological research, but also as platforms for use in human clinical care. But you're really missing a lot of the action that, in the case of neural circuitry, it extends down into the depth of the brain. And you're not really able to engage directly with that part of the overall organ. And so that really motivates the idea of a three-dimensional framework that might be able to overcome that surface integration level of limitation. And so you think about 3D, you maybe immediately think about 3D printing techniques, and there's a lot of sophistication that's been developed in that community over the last 10 or 15 years. And some of the outcomes of that research are illustrated on the right here, starting with the multi-photon lithographically defined truss-like framework to these direct write interdigitated electrodes used for tiny batteries to these various kinds of constructs in the bottom right, defined by high-resolution forms of stereolithography. Great techniques, but really printing of silicon CMOS or transistors sort of outside of the capabilities. So you can print specialized classes of materials. And if you want to create a 3D plastic structure, you're in business. But if you want to do something like construct an optoelectronic system with three-dimensionality, these techniques are irrelevant for that. So then you think about other sorts of approaches, some of which rely on controlled patterns of swelling or residual stress. You can get things to kind of curl up out of the plane, in some cases to create inductors that avoid parasitic coupling to the underlying substrate. Sort of primitive forms of 3D, but not really kind of what we had in mind. We were interested in something that would look kind of like a neural network or vasculature construct, and these techniques really don't get you there. What we had in mind, ideally, you would use this kind of platform as a starting point. So fully processed, state-of-the-art, most aggressive node silicon CMOS, and configure it in some way, perhaps such that upon flip of a switch, the active functional elements on the surface of this silicon would move up out of the plane in some kind of coordinated manner to form a 3D framework with the geometry of interest. And we thought about that, and then had almost decided that maybe that's impossible, but then we found out it's actually pretty easy. And I'll describe how that works in the case of some simple geometries, but bear in mind what we're talking about are techniques that apply to any kind of thin film microsystems technology, not just integrated circuits, but microelectromechanical systems, microfluidic systems, and so on. I'll give you a number of different examples. And so the approaches sort of leverage some of the ideas that I described in the context of the skin, the brain, the cardiac interface, soft electronics, but kind of extend them to allow deployment into that third dimension. And the scheme is shown here as a very simple illustration of the ideas. And so it involves two parts. One illustrated at the top is just a stretchable elastomer substrate. So a silicone polymer formulation, for example, that exhibits linear elastic response to large strain deformation. And that deformation is going to exist in the form of just uniaxial tension in this particular example. So you take that silicone substrate, and you stretch it out like a rubber band up to 300, 400, 500%. And you then functionalize the surface of that silicone uniformly to present hydroxyl termination. And so you can do that just by exposing it to an oxygen plasma. So that's kind of step one. Step two is you take that silicon CMOS wafer that I showed you in the previous slide, and you slice the active stuff off of the near surface region of the underlying wafer. Very similar in concept of what I described previously as a route to construct these flexible silicon nano ribbons. But in this case, it's not straight ribbons. We'll just consider the example of this kind of filamentary structure illustrated in green, where the amplitude of that sort of filamentary pattern decreases linearly in amplitude along its length. So we slice it off, we pattern it in that way, and then we flip it over. And we lithographically define hydroxyl termination at certain locations, illustrated by these red dots. And then when we bring those two things together, this surface functionalized ultra-thin filamentary structure of silicon and that pre-stretched silicone substrate, what will happen is you will get the condensation reactions to occur at all of the regions where you have OH groups coming into contact from both sides. And what is the consequence of that decondensation reaction is strong bonding. So you end up with strong bonding at the positions of these red dots. All other locations are adhered just by generalized adhesion forces, dominated typically by van der Waals interactions. And that's important because it's very weak bonding by contrast to the bonding that occurs at these red dot locations. And so then the final step is you just relax the stretch in the silicone substrate. It snaps back to its original dimensions. And by doing so, it induces compressive stresses on this silicon filamentary structure, but only at the points where the silicon is strongly bonded to the silicone substrate. In all the other locations, what those compressive forces result in is a delamination and a coordinated collection of rotational and translational motions that geometrically transforms that planar serpentine structure into conical helical coil, as illustrated in various angle views in the bottom of this slide. So if you take a look at that process as modeled by finite 3D finite element modeling, what you end up with is something like this. So it's a 2D to 3D transformation induced by a controlled pattern of buckling imposed upon a two-dimensional precursor structure. So that works pretty well. You can use that computational model as a design guide for determining the layouts of these 2D precursors, the magnitude of the pre-strains to generate structures of interest, 3D structures of interest. So on the left, we're showing the results of that finite element modeling, both in terms of the stresses and strains in the substrate, but also in that filamentary construct of silicon. And on the right side, you see a colorized SEM view of a pair of these helical conical coils sitting on top of that assembly substrate, which happens to be polydimethyl siloxane in this case. And so this is device-grade monocrystalline silicon, and you can do that without creating fracture-inducing strains as a result of the geometry transformation. So it's a pretty simple idea, but it turns out it's very scalable, and you can use it to create a very wide variety of 3D mesostructures, starting from planar precursors. Here's an example of a 2D filamentary mesh, much like the mesh structure in terms of geometry for that skin integrated device that I mentioned previously. But here we've bonded it selectively at strategic locations to underlying biaxially pre-strained substrates, such that upon relaxation of that biaxial pre-strain, you induce a geometrical transformation that goes from 2D to 3D, in this case consisting of a parallel array of helical coils as a base layer and a support for another parallel array of helical coils sitting on top of the first and oriented orthogonal to that base layer. And this is what it looks like, colorized SEM angled view of this system. You can see the bonding pads a little bit larger. We can increase the areas to increase the robustness of the bonding to ensure that the structures don't delaminate upon 2D to 3D geometry transformation. We can kind of see how this works, and that's a defect-free example of a sample produced in this way. Here's a cross-sectional SEM view to give you a sense of the extent of three-dimensionality. So not quite a neural network beginning to, however, resemble that kind of three-dimensional open network type of architecture, which again is ubiquitous in biology, absent currently at least from man-made microsystems technologies, with the exception of the constructs that I'm showing you today. So you can do that with ribbons, you can do it with plates, membranes, all sorts of different things. It can be a single layer buckling, multiple layers, hierarchical buckling. Here's an example of a flower-like structure that grows up from a planar precursor away from the underlying substrate as a result of relaxation of that pre-strain. And that can all be captured, again, quantitatively by mechanics modeling. Turns out to be very important as a design guide, but it can be realized experimentally as sort of the key message here. So it's almost like Kirigami, like pop-up book, I guess, is involving some similar types of concepts, but we're applying it here to various kinds of microsystems technologies. And you can actually get even more sophisticated because you can use multi-layered precursors. Doesn't have to be just a single layer, but actually multiple layers. And each one of those layers can be bonded at different locations to the underlying assembly substrate. And so you can create nested sort of space-filling 3D constructs as illustrated in the lower right, or even entanglement can be engineered into these systems. So anyway, many different design parameters, and we're moving toward a system that provides sort of inverse design capabilities. So you can draw the layout of the 3D structure you want, and then the computational tool will tell you what kind of 2D precursor and what sort of pattern of pre-strain you need in order to achieve that 3D structure. So I'm not going to talk about that in any more detail, but just try to describe how these are relevant, maybe as bio interfaces, and give you a few different examples and some biohybrid robots and some bio-inspired microflyer structures as well. So as I mentioned before, you can think about these constructs as scaffolds, but now they're not just passive dumb scaffolds. You have full electronic functionality embedded within them. And so you can seed the growth of neurons and neural tissues around these kinds of scaffolds. And that's what we've done here with rat DRG cells. We seed them onto these 3D constructs, which are populated with electrodes for stimulation and sensing. And now you can grow kind of 3D neural networks, very much similar in geometry, at least, to those that occur natively in the organism. And then you can interact with those 3D networks electrically using offboard electronics. So here's an example of a basket. We're measuring individual firing events, and we can stimulate individual neurons in that 3D type of geometry. You can do that with neural tissues, but also cardiomyocytes, where you have electrical activity and mechanical activity that turn out to be interesting. Here's an example of a different sort of framework. We're using a hydrogel, embedding that framework in the hydrogel and seeding the hydrogel with cardiomyocytes. And so we can watch the spontaneous firing of those cells. We can measure the mechanical activity of them as well. And in this particular case, we have the ability to deliver pharmacological agents on a triggered basis at certain locations in a spatiotemporal fashion as well. So it's one example. The other is thinking about using these ideas in the context of organoid science, hot area of biology these days, whereby human stem cells are differentiated and then allowed to grow into miniature versions of naturally occurring organs. So mini brains, small scale intestinal type structures, kidneys, and so on. And the appeal here is then you can do in vitro studies of the responses of patient-specific artificial organs to various therapeutic interventions and pharmacological agents. Now, the problem is you can examine these types of 3D structures pretty well by optical microscopy, but if you want a hardware interface to them, it becomes much trickier because the traditional scheme for studying tissue cultures in vitro involved 2D arrays of electrodes, multi-electrode arrays, and that 2D geometry just doesn't match the intrinsic geometry of these organoid constructs. And so we thought about creating 3D frameworks that could sort of softly envelop the entire outside surfaces of these organoids, much like that cardiac sock that I mentioned to you before, but now on this sub-millimeter length scale, to allow for the delivery of various kinds of sensors and actuators to the outside surfaces of these organoids to probe their properties and stimulate responses within them. And so here's an example of that. So we can exploit all of the sophistication in 2D material science and lithographic processing that's been developed in the semiconductor industry over the last half century, and develop really sophisticated forms of electronics in planar geometries. And as a final step, we do this buckling process to transform that 2D construct into a 3D framework, shape match to an organoid of interest, which in this particular case consists of a cortical spheroid. So it's a spherically shaped mini brain with a diameter of about 0.5 millimeter. And so we create a basket structure that consists of this collection of ribbons. Each ribbon is populated with various kinds of sensors and actuators illustrated in the micrographs in the bottom right part of this view graph. So you see microelectrodes, electroplated platinum black, for example, cellular scale LEDs for optogenetic interfaces, thermal actuators and precise temperature sensors to look at metabolic action. Various kinds of electrochemical sensors as well can be embedded in these frameworks. So now you have a 3D basket. You may wonder, how am I going to get my spheroid down into that basket? Well, you can just exploit the fact that the 2D to 3D geometry transformation process is purely linear, elastic, and reversible. So after you form that basket, you can open it up by locally deforming the underlying substrate to create that opening, and then drop the spheroid in at that point. And so this is 3D finite element modeling of that opening up process, and then you simply drop the spheroid down into that structure, and then relax that localized strain field in the underlying substrate to allow that basket to then enclose the organoid in a slow, gradual, non-invasive processing. You see the sequence of optical micrographs in the lower left showing the organoid placed in the partially open basket structure, and then watch that basket gently enclose the spheroid and deliver those electronic components then softly and gently to the surface of the spheroid, because the system is designed using that finite element modeling tool to shape match the organoid of interest, in this case cortical spheroid derived from human stem cells. So that's what it looks like. What can you do with that? Now you can do 3D electrophysiology. So you're sort of doing micro electrocorticography, but now on a mini brain structure around what is sort of the vast majority of the outer surface of the sphere. Not 100%. You're missing sort of the cap structure, but pretty good coverage. And the structures themselves are sufficiently optically transparent. You can do simultaneous optical microscopy. And so this is a rendering of random patterns of firing across the surface of an organoid in this 3D type geometry across 30, 40 electrode measurement sites in this particular case. So what's a specific application? We're working with folks in neuroregeneration and rehabilitation to understand how neurite bridges form and how healing occurs in neural tissues. And so to do that, we exploit sort of the versatility in 3D geometries to create a framework that holds not just one spheroid, but actually two spheroids. And it holds two spheroids in close proximity to one another. And what will happen is that over time, a neurite bridge will begin to form between these two spheroids, A and B. And you can kind of watch that happen by optical microscopy, but you can't determine just by microscopy whether that neurite bridge is functional or not. But with the electrodes, you can begin to look at the functionality of that bridge by searching for coordinated firing events that are observed in spheroid A and spheroid B as an indication of the functional formation of that bridging structure. And so you can watch that bridge form, you can watch the coordinated firing events occur, and then you can come in with the scalpel and you can cut that neurite bridge away. And then you can destroy that coordinated electrical activity, and then you can watch it reform over time. So we think this is a very powerful platform for studying fundamental processes in neurodegeneration that are relevant to traumatic brain injury. And we're doing that with experts at the Shirley Ryan Ability Labs, very large rehabilitation hospital in Chicago, just to give you a sense of what's possible. So you don't have to stop there. You can think about even more elaborated frameworks that can hold more than just two spheroids. Here's an example of 3 spheroids. So now you can think about using these spheroids as like building blocks. You use neural, muscular organoids and begin to put together functional systems, maybe xenobots one day, and really co-integrate electrical functionality within those constructs. But one question you have to ask is, as these get larger, just passive diffusive transport of waste and nutrients is insufficient to support cellular function at the cores of these structures. And so you end up with a necrotic core once you get above length scales of about 1 millimeter. And so then you would like to think about artificial vasculature that you kind of embed in these structures to avoid that kind of necrosis. And a lot of the ideas I've already described in 2D to 3D transformation of electronic systems apply equally well to microfluidic systems. And so we can create space-filling constructs of arteries, arterioles, capillary beds, and so on to overcome that problem of sustaining life in larger scale engineered biological tissues. And so this is some examples of those 3D microfluidic structures as a direction that we're going. But you don't have to just think about neural spheroids and organoids. You can think about other systems as well. This is an example of a cardiac organoid that we're working on with an expert in cardiac science, Beth McNally. And so she can grow these artificial sort of cardiac rings. And here it's mounted on a 3D construct that provides electrophysiological monitoring capability, but also has the ability to measure forces. So there are strain gauges embedded in each of the ribbons in this sort of structure. And so we can look at coordinated electromechanical behaviors of this cardiac organoid. And so we can do ECG essentially at multiple sites across that cardiac organoid. And it looks like my PowerPoint has crashed. So let me just try to restart that. So you can monitor ECG at different spatial locations across that ring. And then you can measure propagation velocities associated with those waves of depolarization. And let me try to restart this. Give me 1 second.
No worries at all. Take your time.
Yeah, it'll only be 1 second here.
No, but PowerPoint has a mind of its own.
Yeah, I don't know what's going on with my computer. It's the second time this has crashed.
Oh no!
In the last few days, so that I'm back in business. Hold on.
Oh, excellent. Thank you.
Yeah. So let me actually let me zoom to that position. And so that got us interested in thinking about mechanical properties and embedding mechanical actuators, not just mechanical sensors, but structures that could actuate motion. And again, these ideas in 3D are compatible with any kind of planar materials technology. So we can immediately integrate piezoelectric actuators, and we can localize those actuators at different positions so that they can independently be actuated to excite different normal modes in these 3D frameworks. So in this case, sort of a table structure. And so we can actuate each one of the legs, different phase and amplitudes, and we can actuate the table as well. And so not just that kind of table structure, but all kinds of 3D frameworks can be excited into normal mode motions. And these are finite element modeling results and experimental images of different structures that we've looked at. And then we're kind of thinking about this. It looks like these things would want to walk away, or in the case of the upper left case, maybe fly away. Maybe you could flap these wings fast enough, it could take flight, or you can make them walk fast enough they could move away from their initial locations. The only problem with that is that based on the 2D to 3D transformation process, if these adhesion sites become delaminated, then the overall structure just pops back into that original planar geometry. So that's problem number one. A lot of other problems as well, if you want to think about isolated robots, but that's problem number one. So the question became, how can we freeze the 3D shape into that geometry so that after it's removed from that underlying assembly substrate, it would hold that geometry. And there are a variety of ways to do that. It turns out the simplest and most versatile is to use shape memory polymers. A lot of polymers have this property, and so you can buckle them up into a 3D geometry. You heat them above their transition temperature and then cool them back down. It fixes the shape. So now they can be released from the substrate without popping back into that original planar geometry. And so that turns out to work really well. And then it suddenly opens up the possibility of creating 3D frameworks that could be the basis of various kinds of robotic structures, terrestrial robots, swimming robots, or maybe even flying robots. And they could be designed just out of man-made material systems, or they could be co-integrated with active biological tissues. And so that's what we got interested in and started working in that direction. Here's an example. I think this is the smallest terrestrial robot ever developed. It's about 0.5 millimeter on a side in geometry of a crab. Students wanted to build a crab, so they did that. And we have the actuators are based on shape memory alloy. And so we have actuators at the joints of each of the legs. The claws don't do anything, and neither do the eyes. It's just for aesthetic purposes. But anyway, you create these things. You do lots of them in parallel. And then you scan a laser beam across the body of this structure. That induces local heating and a shape change associated with the shape memory alloy. And that provides the basis for controlling the gait and locomotion in these robots. And so here's an example of one of those crabs moving along, excited and controlled by a scanned laser beam across the surface of the crab. So bio-inspired, I guess, at some level, but you don't need to constrain yourself to what exists in biology. There's all kinds of different 3D structures that you can actuate in this way: walkers, jumpers, crawlers, different kinds of geometries. Here's an example of something that kind of hops along. This is at 1x speed, so this is real-time speed. You can see the scanned laser moving across, and it causes a sort of a jumping motion every time the laser beam interacts with that structure. Sort of like grasshopper or cricket, but with much different underlying physics in terms of how the motion is occurring. But then we thought, well, we're doing these cardiac organoids. What about muscle rings and so on? And you get away with the shape from the shape memory alloys, and then integrate sort of active electronic functionality, optoelectronic functionality to trigger the contraction of the muscles, and then create a biohybrid robot in that way, using the 3D framework as a skeleton, but also as a means for control and communication. And so that's what we've done here. And this was published very, very recently, just to give you a sense of what's possible. And they can do like basic work of various sorts. You can have them push stuff around. And these are muscle rings, in this case, optogenetically controlled in terms of their actuation. So it looks like I'm crashing again, which is not good. But this is kind of the sort of thing that you can do and think about biohybrids, not only from the standpoint of muscle rings, but also integrating neural tissue and establishing neuromuscular junctions. So you control the muscle from the neural tissue rather than direct engagement with the muscle tissue would be is something of interest and something that we're working toward currently. So this is one more crash, and I think I'm going to be done in 2 more minutes. So hopefully we can get this done with only one more restart. Let's see how this goes. So that's what we're doing, around walkers and biohybrids and bio-inspired systems. But then we didn't want to let go of this idea of flyers, things that might be able to take flight and move in that sort of mode of locomotion. And so let me just conclude with 2 minutes on what we're doing in flying 3D structures, here relying very heavily on bioinspiration. So we wanted to create like a gnat or a fly, but that's too hard because it requires power supply, guidance systems, and so on. And so we looked to nature to think about maybe a simpler starting point, and we got fixated around wind-dispersed seeds. There's really spectacular aerodynamic designs built into seeds of various sorts, where the evolutionary forces are driving toward mechanisms for dispersing genetic information at the over the longest possible distances to propagate the species. And so there are different kinds of flying seeds. And so we wondered about the ability to deliver electronics across a large environment for purposes of environmental monitoring. And specifically, could we add winged flight to microsensors and microchips and radios and things like that? And could we use some of these techniques in 3D fabrication to sort of do that? And we thought about gliders and parachutes, but then we thought it would be more interesting to think about helicopters. And so maple seeds and tristillaceous seeds, they fall in this way. And I think the motivation from an evolutionary standpoint is to reduce the falling velocity, the terminal speed, to increase the probability that random patterns of wind can disperse the seeds. And so that's the idea. And so we got started on this and used a lot of modeling and different schemes to design winged structures that would optimize that kind of rotational sort of dynamics during free fall. And you have a tremendous amount of versatility in the attack angle, the shapes of the wings, the number of wings, the extent of three-dimensionality. You can add porosity, all sorts of things become possible. And you could sort of explore this design space almost in a combinatorial fashion because, again, the 2D to 3D transformation process happens in parallel. So you can generate hundreds of different types of geometries by planar processing and then pop them up into 3D, fix their shape, release them, and then drop them and then study their aerodynamics as a function of geometry and size and weight distribution as well. So very tiny structures is kind of where our interests are because of the cost considerations. Smaller is cheaper in microelectronics. And so adding wings to very small microchips becomes appealing. And anyway, you can kind of do this. And the key performance metric for us is terminal velocity. This is an example of terminal velocity in one of these 3D microflyers compared to other sorts of objects you see in biology. So I think I'm almost out of time, so I'm just going to skip ahead and save you, all the details around the aerodynamic studies and try to give you a sense of what we want to do with this. I think there are opportunities in environmental monitoring, so large spatiotemporal monitoring of processes in the environment by distributing tens of thousands, maybe hundreds of thousands of these tiny sensors across an environment and then reading them out remotely. And you can think about reading them out electronically, but you can also use high resolution digital imaging techniques and colorimetric responsive chemistries to do the readout in a more passive fashion. And you can build these colorimetric flyers out of bioresorbable materials, so you don't have to worry about polluting the environment or recovering the devices after they're deployed into the environment. So we can do that. And then you do the readout with a drone is the idea. So you distribute them with a drone, and then you do readout at various times by flying the drone over the area of interest. And we demonstrated this in very simple pilot scale study. We're using high resolution digital image capture and color extraction techniques to determine the response of these flyers to local environmental parameters of various sorts. So with that, I think I'm going to conclude. I'm out of time. I apologize for the computer issues, but hopefully some of this content is of interest and synergistic with this overall meeting. So we're interested in 3D, and I talked about neural interfaces, cardiac organoid interfaces, bio-inspired robots, and bio-hybrid robots as well. And so I'll just conclude by acknowledging senior collaborators in engineering science, medical science, very important for us. But the most important people are the students and the postdocs who do the work. So I always conclude my talks by acknowledging them for their hard work and their great ideas, and thanking you for your attention.
Thank you so much, John, for a really inspiring talk, touching on so many areas and so many interdisciplinary aspects. Really, really fascinating. So thank you so much for your time. Now, I'm aware you have to head off quite soon, don't you? So I think we've only got a few minutes for questions. How long do we have? 5 minutes? Is that
sure?
2 or 3
sure,
minutes?
5 minutes
Okay,
is fine.
perfect.
Yeah.
So just one question I have is, you approach a lot of this with FEA or simulation, and also even when you're putting things in vivo, do you consider simulation of the surrounding material, or how accurate is that simulation, or is it a limiting factor for what you're doing?
Yeah, that's a great question. Most of the modeling that we have done has been focused on the 2D to 3D transformation process itself, and that occurs in ambient conditions in open air, so we don't have to worry about the effects of the surrounding environment on the transformation and the final 3D geometry that results from that. But I think you're asking about whether we do modeling of how the biological tissues engage with those 3D structures. And we've done a little bit of that as well. One of the challenges with these organoids, they're extremely fragile, super low modulus, like 5 kilopascals or something like that. And so, for example, when that basket structure begins to enclose upon the organoid, it doesn't take much to sort of deform the organoid if the framework is too small. It's not fitting the geometry and the diameter of the spheroid. So we have thought about things at that level. In terms of the cardiac contractions, we designed the frameworks not to impede in any significant way the strains that are induced by the contractile behavior of the cardiomyocytes. So we do a little bit of modeling there, as well as just to understand how the strain gauges are responding to the forces imposed upon them by the cardiac muscle tissue. So bits and pieces, but probably we could do a lot more along those lines. I think maybe that's where your question is going. So it's probably an area of opportunity to explore that further.
Thank you very much. And one other question just about the inspiration behind the design. So the last on the flyers you presented with bio-inspiration, but some of the other structures, where does the inspiration come from a lot of your work? Is it rooted in kind of trial and error, human intuition, bio-inspiration? Is there a specific methodology you use to come to these fantastic ideas?
Pretty chaotic, I would say. I don't know. I kind of get into brainstorming sessions with collaborators who want to do something with these 3D things, a little bit of give and take there, and a little bit of just wild ideas. As an academic group, we can do that sort of thing, crazy stuff. So yeah, all of the above, I'd say.
And just a couple of quick questions from the chat before I know you have to go. One was Professor Rogers, as usual, great talk. They were interested to know the role of biofouling on the interface of these materials. Any strategies to avoid it, and would the surface chemistry functionalities on these materials affect mechanical properties too?
Yeah, it's always a problem, be honest with you. A lot of the fouling mechanisms are poorly understood. So we don't have any magic in that sense. Although I would say that in terms of neural interfaces, historically, work in that space has been dominated by electrical interfaces. So electrode surfaces in direct contact with neural tissues, and you pick up electrical activity in that way, or you deliver electrical stimulation. Their biofouling is a big problem, especially in the sensing side, because it increases the impedance and it reduces the signal magnitudes. For stimulation, you can kind of get away with just increasing the voltages and the currents that are being delivered. So you just plow through the biofilm and you kind of do things that way. But my feeling is that optics is the future of neural interface technology. And with optics, a very thin biofilm, as long as it's sufficiently transparent, doesn't matter. And so that's where we're headed. And maybe that's a cheap way out. We're not going to solve the problem. We're just going to avoid it. That's the thing. So most of what we do in neural interfaces is built around optogenetics and genetically encoded markers for measuring calcium and so on. And it's a tremendously active field. I think it has to be the future. Some people are a little bit nervous about getting into it because a lot of those techniques have not been approved for human use. And that's understandable then. It's still very valuable for animal model studies, but my guess is it will receive approval. It's just a question when that will happen for humans. And once it does, I think it's going to be optics. I mean, maybe I'm wrong, but that's the bet we're placing. And so we just sidestep this thorny issue of fouling.
Very, very good answer. And just last question, also from the chat, could the 3D structures form pumps to cool microchips?
Yeah, so I didn't talk about that, but we're very interested in thermal modes of engagement as well. Also, don't have to worry about biofilms with thermal either. So we have ways to do targeted evaporative cooling. We published a paper recently on nerve block using a bioresorbable cooler as a way to treat acute forms of pain. And we're integrating that same kind of micro cooler technology into these cardiac organoid rings, because you can silence the activity of the cardiomyocytes by cooling them. And that opens up some interesting opportunities in biological research that we're pursuing with our collaborators. So I really like cooling. I think it's an interesting direction, and it's largely unexplored to this point because of just technical difficulties of how you deliver cooling power locally to a region in deep tissue. It's hard to do that, and thermoelectrics don't work too well. So we're very interested in that angle.
Brilliant. Well, thank you again, Professor Rogers. A really inspiring talk, and so relevant to a lot of the topics we've been discussing. So thank you so much for your time. Sorry about the confusion on the hour difference, but we'll let you go. So thank you once again.
No problem. Thanks for the invite. Yeah.
Thank you.